So it's a great pleasure to welcome you for the overview about work package 3 on advanced multicellular lung models and how we use them and optimize them for nanomaterial hazard assessment. It's a great pleasure to do this final presentation together with the co-leader of this work package, Martin Klicht from Swansea University. So let me start and thank everybody in work package 3. It was such a great pleasure to collaborate, and of course I have very mixed feelings. It's fantastic to show the achievements from the previous 4 years, but I'm also very sad that the collaboration now will stop officially end of September, and I hope that we can continue some of the collaborations afterwards. I would like to start with this nice sentence from David Brown from Heriot-Watt University, and I think what he says is really true for all of us. We could never have such a great collaboration and achievement without this great team spirit we had during all these 4 years. So let me start to give you a quick outline about what we will present you. First, we will focus on systems and show you the lung model development and exposure approaches. Then we will show you how we used the optimized systems for nanomaterial hazard assessment. And we will close with some lessons we have learned and take-home messages. So at the beginning of PETROS, we started to compare different bronchial and alveolar cells, mostly focused on cell lines. We cultured them on these in these transfer systems, which enabled us to expose the cells at the air-liquid interface. And I just give you some examples of cells that we started to compare: Calu-3 cells, A549, Helvy cells, and the focus of this work was really to check how stable the cells are at the air-liquid interface, because this is really a prerequisite if you work with lung cell cultures. So I show you one example, which are the Calu-3 cells, and also show you the endpoints on which we focus to characterize the models. So the Calu-3 cells have shown to produce the highest and most stable transepithelial electrical resistance when they were cultured for several days or even weeks at the air-liquid interface. There is almost no cytotoxicity reported during these 3-week cultures. And what is really important is that they are stable. They show a nice monolayer even after 2 months at the air-liquid interface. Another important endpoint was to show that the cell models are responsive to positive controls such as lipopolysaccharide. Already at the very beginning of the project, we started to compare the protocols among labs, and this is a study which was performed by my lab and Martin's lab in Swansea, where we used A549 cells, so a rather simple cell systems, and compared submerged and air-liquid exposures. We exposed the cells to lipopolysaccharide and TNF-alpha. And here you see some of the endpoints in orange, the data from my lab, and in blue, the data from Martin's lab. And you see there is quite a difference for some of the endpoints. However, the final conclusion was that the tendency is the same, also in regards to the positive control treatments. And we already could show that with a simple monolayer system, it's very important to harmonize the protocols and also to use, if possible, the same cell culture consumables. And this information was then also important for the last phase in PETROS. So each step in the project was really discussed among all the partners and collaborators. And only with this we could produce some of the SOPs, and also we could agree on how we approach some of the questions, and one is the co-cultures with the macrophages. So we spent quite some time to look into the number of cells, so the ratio of epithelial cells and macrophages, and we also developed a strategy for the prolonged lung cell cultures by adding macrophages every week to the epithelial tissue. We decided to focus on two macrophage systems. One are primary macrophages, monocyte-derived macrophages, and the other one is a human cell line, the THP1. And we developed a standard operation protocols for both macrophage types and for some of the co-cultures. Another system which we started to use to integrate was the epialveolar system, which in my lab has been already developed in collaboration with MATE Corporation and PETA International Science Consortium. And here we increased the complexity. Here we used primary epithelial cells, alveolar type 1 cells, fibroblasts and endothelial cells. And when the cultures were shipped to our lab in Fribourg, we added macrophages. And one of the first tasks was to show that the cell model is responsive to transforming growth factor beta, which can induce a fibrotic phenotype of the cultures. And I think it's also important to highlight that it's not only a multidisciplinary teamwork, but we also come from very different disciplines to address some of the challenges in petros. And one of the great new collaborations we had was with Professor Artial Balia from Uni Pisa. And they came into the project because they have developed the DALI bioreactor. So this is a bionate membrane, which you see here on the left side. And the great advantage of this membrane that it is flexible, so it can be assembled in a bioreactor, which you see on the right side. You can induce the breathing, stretching of the membrane, and in the lower chamber, you introduce a flow, so you mimic the blood flow in the alveolar region. So here on this video, you see this stretching of the membrane. Below you see the cell culture medium, which is flowing in one direction. And in my lab, we have then started to look into A509 cell growth. This has also been done in Martin's lab, so we could transfer the DALI bioreactor into two labs participating in petros. Here the work is still ongoing. It has been shown that it is very challenging to grow the cells on this bionate membrane, and we will see more tomorrow from the presentations of the young researchers.
Okay, so moving on, once we'd got our systems up and running and characterized, then we wanted to start to implement the tier 1 patrols, engineered nanomaterials, as well as positive particle controls. And so having these advanced systems that we have, as well as the monocultures, we want to make sure that we continue to go towards or achieve the realistic scenario that we want to have. So with that, we adopted a variety of different exposure approaches for the engineered nanomaterials and positive particle controls. One of these was the quasi-alley exposure, which is in between aerosolized cultures and submerged cultures. So the cells are still at the air-liquid interface, but they have a small volume of liquid, 100 microliters to be exact, carefully pipetted over the top. And to compare that with the now well-known VitroCell Cloud 2 or VitroCell systems, where we can aerosolize liquid exposures onto the air-liquid interface cell culture systems. So we've done that, but then we also wanted to start to implement the different kinetics of exposure, because commonly a lot of research is just focusing on a specific time point or just a single exposure. And there's a movement now within the literature, as we'll go on through in later slides, that highlights that the repeated exposures with low concentrations is more realistic towards the real world scenario. And so for that, using the short-term system that Barbara's already highlighted, using A549 cells with DTHP1 macrophages on top, we looked at different exposure approaches at different concentrations with collaboration through work package 6, where we understood that these were extrapolated from in vivo scenarios. that we either did it over 24 hours, 48 hours, or 72 hours. And this was either done as a single or repeated exposure. And what we found when we looked at a specific set of endpoints, specifically pro-inflammatory mediators and micronucleus assay to assess genotoxicity, we found that actually 48 hours repeated for the variety of different materials that we exposed in this small study was the best way forward and gave the best sensitivity of this model. Of course, using liquid exposures is one way to do it, but predominantly what we inhale is a dry powder, especially in an occupational setting. But work done by David Brown at Heriot-Watt University over the course of the project just showed how technically difficult it is to get a good repetitive and reproducible dry powder exposure approach to cell culture systems cultured at the air liquid interface. And so there's been a great, okay, so now we're just going to move on and we're going to understand how these systems can be used to deduce ENM hazard assessment. So first of all, what we did is we focused on the short-term culture, so the A549D THP1 co-culture again, and we assessed this against a specific well-established adverse outcome pathway, so specifically AOP173, as you can see at the top of this slide, using titanium dioxide and zinc oxide nanomaterials, as you see here on the slide, at in vivo extrapolated concentrations over 6, 24, and 72 hours. We assessed a specific gene list through PCR array technology to understand whether or not this system can actually indicate similar responses as seen in vivo. This approach was also adopted in a collaboration with Health Canada, where we've undertaken transcriptomics work with them to get a more global understanding of whether or not the predictive quality of this system is true or not. Moving forward with the global understanding of the predictive quality, or even the understanding of the toxicology of these nanomaterials with the short-term system, in collaboration with MISFIC, we've done a bit of work to deduce whether or not our advanced lung cell culture can be used inside of their technology, specifically their lysate microarray technology. And so there's been a lot of work done to try and understand this method, because commonly it's Vesser at MISFIC is using single cells, and he's exposing the cells into the engineered nanomaterials suspension. And since we expose our cell cultures to the nanomaterials in a different way, we wanted to understand whether or not we could actually achieve the way that we do the exposures, and what, as you've seen with the amount of work that we've put towards that, can then be fit into their lysate microarray approach. Unfortunately, we've got no data to show you of that, because even though the UK to Finland is only a 3.5 hour flight, the inability of couriers to provide the samples in good time and without being thawed was a constant thorn in our side. And so this is where we're having to sit at the moment to determine how we can go forward with that. But not to say that MISFIC didn't do anything else. They did a lot. They also conducted a lot of monoculture analysis on an array of different engineered nanomaterials, predominantly Petrol's tier 1 materials, but also other ones, as you can see here on the far right-hand side of this slide. And so they gave us a good understanding of how these materials relate to one another, and can put that into context, as you see in the center of this slide, as to their toxicology and their ranking. Next slide, please. So moving on from the short-term exposures to the relative long-term, we move on to the work from Sybil and Dominique at LTAP in Belgium. And predominantly they looked at fibroblasts and 2 different fibroblast cell types. And they looked at short-term and they looked at long-term. And you can see their experimental approach here on the left-hand side. And what they did is direct and indirect exposures. And for that, direct is exposures directly onto the fibroblasts, and indirect was through conditioned media exposures from samples produced from either the short-term exposure system that I just talked about, or the long-term that Barbara's already highlighted, and we'll go into in further detail in the next slide. But overall, what they found was an array of data, and too much to go into detail at this moment in time, but the key finding was that they saw that OPN was a key marker within the fibroblast cultures of understanding the fibrotic nature of any engineered nanomaterial, particularly carbon nanotubes.
So as Martin already highlighted, we were able to move from short-term to long-term nanomaterial exposures not only with quasi-Ali approaches or fibroblast assays, but also with the lung cell models we developed in the first phase. And for this, we defined some scenarios to mimic daily repeated exposures, which is really relevant in an occupational setting. And here I would highlight a great collaboration with work package 6, which recommended us concentrations of the nanomaterials which are in correlation with human or animal data, where they have shown that with this concentration, some of the endpoints we are interested in, like inflammation or fibrosis, is occurring. So we used then these concentration recommendations and started with the cloud system to expose cells to one concentration or with repeated exposures to higher concentrations of the nanomaterials. Here just an example of silica quartz, nanosil, and Mitsui carbon nanotubes. And we have then used the epialveolar tissue model, which is stable at the air-liquid interface for several weeks, and started to apply the adverse outcome pathway for pulmonary fibrosis. So we started to investigate if the nanomaterials interact with our cell models, which we showed and proved. Then we looked into pro-inflammatory, pro-fibrotic responses. Here just an example on how such a data set is looking like. So we investigated the cytokine release on different days during the exposures. And we also looked into the extracellular matrix accumulation, which is really one of the relevant endpoints for the development of fibrosis. And we could show that the epithelial tissue thickness increased after repeated exposure to silica quartz and Mitsui 7. And with all these methods and systems established, we then started to move into the inter and intra-laboratory comparisons. And for this last phase, it was really important and a great opportunity to have industry involved, because it's important for us that the systems will also be used by various stakeholders in the future. And here I would like to acknowledge the collaboration with Barbara Birk from BASF.
Yeah, it's a fantastic collaboration by all partners in this interlaboratory round robin activity that was led by Hedvig and Rob at RIVM. And it actually included not just patrol partners, but what we refer to as non-patrol partners across Europe. And it was a fantastic interaction. And what we were able to do prior to any data collection was actually create two very nice SOPs that went through rounds and rounds of iterations, and I think still could now go through rounds of iterations to get the absolute finite detail, which is what I'm sure you can appreciate is key towards experimental harmony. And we can see that when we look at monoculture, we look at the co-culture systems with the different exposures, that within each partner's lab, that the data is very much repetitive and reproducible. But when we start to look across the different labs, that whilst the trends might be similar in terms of the specific exposures, that there is a slight variability in the outputs or the level of the different endpoints that we've assessed. And so this is a good finding, because we didn't want to find that everything was exactly the same, because that would be relatively boring. But it's good to see that the SOPs could be reproduced within the labs, and the differences that we see across labs are still being deduced at this moment in time. Okay. So now for this final part of this presentation, we'd just like to go over some lessons learned and some suggestive take-home messages from our research over the past 3.5 years. So we have some achievements and we have some challenges. The achievements are that we've managed to understand and characterize a range of different lung epithelial cell types from different regions of the human lung. We've been able to culture these both short and long term and use them within hazard assessment of engineered nanomaterials. We've also been able to deduce that single and repeated exposures, depending on the specific culture type that we're using, are highly relevant to deducing that hazard of ENMs and relevant to the in vivo scenario. The challenges that we have had is that some cells won't grow, or some cells won't grow long enough. They're not happy enough, and that's for a variety of different reasons. Also, that we've had a lot of variabilities across labs, and that's actually been a nice challenge to have, because that's enabled us to discuss and get to the finite detail of what the experimental approach was. Next slide, please. In addition, when we're putting together the different model systems, we found that beyond the static controls, being able to use physiologically relevant culture methods, such as fluid flow, dynamic breathing, and also, in fact, just using more primary cells that we can enable the long-term ENM exposure, have been highly advantageous and are a key output from this work package within patrols. In addition, the exposure and the ability to expose cells at the air liquid interface to dry powder scenarios has been a key and impactful study and output, which moving forward. The challenges that we faced, again, with the exposures is the technicality, again, the interlaboratory comparisons, but also the predictivity of the specific key events within the cell cultures. Sometimes the markers that we thought would definitely appear never appeared, but then other markers appeared that we never thought would. And I think this is just key, as we know within science, this occurs. And finally, what we'd like to just say is that we think that the efforts within patrols have resulted in a lot of key data, impactful data, and data that we can use as a foundation moving forward in producing predictive models for the replacement of in vivo research in the future. These models are repeatable, they were harmonized, but of course there's always room for improvement, and that improvement we feel could be done by on-site training. Of course, this hasn't been possible for the past couple of years or 18 months because of the pandemic, but the online webinars and discussions have been advantageous, but nothing beats face-to-face lab exchange of information and knowledge. The availability of the same cell cultures at the same passage or the same batch, and same with the chemicals or the assay-specific aspects, have been a challenge and will continue to be a challenge moving forward, but that's something that we can try and find a solution for. And obviously the SOPs, as I've mentioned, was great to have the extensive discussions, but of course these can constantly be manipulated and improved, but understanding where to draw the line is something that we'll have to deduce as we go along, so that we can actually start to get the experiments and start to get the systems moving forward. So with that, as we've highlighted throughout this, nothing could happen without teamwork, and as Hedvig's highlighted, without all of the experience and the enthusiasm, not just from the senior researchers, but mostly from the young researchers within the work package, to push the research, to ask the difficult questions, even if the answer wasn't clear, was the most enjoyable part, and probably the reason why we feel that this work package was such a success. So with that, thank you very much. These are just the pictures from the very beginning until the very end. And thank you to all, and thank you to the work package colleagues, to Barbara as well for leading the work package so well, and clearly to Shireen for coordinating it so well, and to you all for your time and attention. Thank you.
Thank you, Martin. It was a great collaboration.